Hello everyone. For our first story, scientists can use CRISPR to track cancer growth. Dmitry Petrov from Stanford was able to attach short segments of DNA to tumor cells to act as barcodes. Every time the cancer cells multiply, the amount of barcodes multiply as well, so how much the cancer has grown can be determined. This method makes mapping cancer much easier, which is an important development if we are to find a way to cure it. For the next story, CRISPR is now able to edit genes much faster than before. Scientists at UCLA have developed a way to use CRISPR to edit tens of thousands of genes at the same time. The reason that it was so slow before was that each separate cell needed a specific guide RNA. The new method allows thousands of guide RNAs to be attached to each patch, which makes sure that the correct guide and patch combination will be delivered to each cell. This now allows the scientists to determine which cells affect the genes and which ones do not. The final story is about cell-free CRISPR. A team from the Gene Editing Institute in Delaware used CRISPR-CPF1 to isolate DNA plasmids, which are DNA molecules usually in bacteria that can replicate independently of a cell. Scientists can store the plasmid in a petri dish or a test tube and perform research on it so they can replicate mutations faster than with a normal cell. Thus, cell-free CRISPR. CRISPR-CPF1 is an alternative to CRISPR-Cas9, which has several apparent advantages over it, including the editing speed and the fact that its cuts are much easier to attach DNA to. I've talked about it before in one of my videos, and it seems likely that CRISPR-CPF1 could become the default protein used with CRISPR. Anyway, that's all for now. See you next time. CRISPR Man, out.